So if anyone has questions on that, on how to get involved with programs like EQIP or CSP, you can let me know. I'm kind of the link between the Conservation District and the NRCS, and the NRCS has some great technicians, and we have a great district conservationist there as well. So you can get a hold of me. My email is on this slide, and my phone number as well. So I look forward to getting to know everyone, in another format, and today as well. So today's Shop Talk is a presentation that comes from the farmer-led Watershed Conservation Group. So it's a farmer-led conservation group made up of farmers and technicians like myself who are here to promote conservation, help farmers get involved with MEAP, the Michigan Agriculture Environmental Assurance Program. And it's really to just encourage conservation through a lot of ways, through educational programs like this, and small shop talks, bigger field trips. So that obviously COVID kind of changed that a little bit, made it a little harder, but as things shift towards a little bit of normalcy, hopefully we'll have some more of that happen. And this is also presented by the Washtenaw Conservation District. So we work with landowners on everything from resource concerns to, like I said, in my department, the Farm Bill programs. We have the MEAP program. We have a great tree and shrub sale. So that's kind of a little bit of an introduction. At the end, there'll be a survey, and I'll remind you to take that survey. So let's move on a little bit to my next slide. So I just want to go over a few real quick housekeeping notes. Generally, when you enter a meeting, your microphones are muted. They're not on this. So just make sure your microphones are muted. Everyone will have an opportunity to talk. That's kind of what we want. We want to have some conversation. But wait until that opportunity presents itself. Pretty soon we'll go into breakout rooms, assuming that works. And what we'll ask you to do is to just raise your hand physically like this, and then the moderator will see you. So raise your hand, and then they'll call on you. Don't exit the breakout room when you get into it. When at the end of the session, you should be redirected back to me and the main presentation. And then throughout the shop talk today and our presentation that we will have, which I'll talk about in a minute, feel free to ask any questions just by chatting in the chat box. So there'll be time set aside at the end of the presentation for questions. So that's about it. We're pretty lucky to have someone I've been trying to get involved with going back to my old job by the name of Joe Scrimger. He's a pretty smart guy when it comes to soil biology and kind of a unique approach to agriculture. So we're going to talk a little bit about, agriculture on a bigger scale, and then in a way that's kind of relevant to all sorts of farming. But we really want to build camaraderie in this group of people that we have here today, as well as throughout the Western Lake Erie Basin. So we'll go ahead and get started. And if you're on a phone call and you want something to say and you don't have your video enabled, you can send me a message and we'll get to that when the opportunity itself. So anyway, we're gonna see how this works. So we're gonna try something very unique here, which is a breakout room. And this is just our little agenda for today. So I'm gonna do a quick breakout room. We're gonna talk about conservation, a few questions to see where people are at. The moderator is gonna report back to the group, and then we'll have our presentation. So let's see if my magical powers work here. Breakout rooms. Open all rooms. Okay, so let me know you should be redirected.
You've got to press join It'll pop up on your screen, right?
Yeah, so just hit join and you should be.
Hi, everybody.
Hello, Megan. Hi, everybody.
This is fun.
Yes, it is.
We're in a pop-out room, a breakout room. Welcome.
So far, so good. It worked, right?
Yeah. So I think we'll have 10 minutes total. So it's sort of a lightning round, which is exciting and stressful at the same time. I'll just introduce myself. I'll be the moderator for a little breakout session. My name is Megan DeLue. I'm the Executive Director of the Conservation District here in Washtenaw County. And I'm also a farmer in the River Raisin Watershed over in Manchester. So maybe since we're a small group, just real quick, say your name and where you're farming at. So Eric, you want to go first?
Sure. Hey, everybody. My name's Eric Campy. I am farming just outside of the city of Ann Arbor. I'm involved with the Green Things Farm Collective and Ann Arbor Seed Company. We're growing produce, seeds, a very small herd of beef cattle, flowers. I don't know, everything we can do.
Thanks, Eric. Lori Phelan?
I am Lori Phelan, and I am with Michigan Association of Conservation Districts. I'm kind of an outsider here. I actually am a property owner and farmer in Berry County, but I really enjoy working with all of you folks in the WLEB, and hopefully I can bring back lessons learned from all of your work to my home county as well. Thank you.
Thanks, Lori. Mary Fowler? You're muted, Mary. There we go.
I'm a master gardener. I live in Ann Arbor, and I do a lot of gardening, landscape type stuff. I'm here because I grew up with two families of farming, and I'm not seeing conservation where I live, and I think there's a need for it.
Thank you, Mary. Joe, you want to give us a preview? Introduce yourself?
Yeah, so not currently farming. I get to do a little bit with the cattle on my daughter and son-in-law's farm a couple miles away. But we moved down here to Battle Creek a couple years ago from the Thumb, and we farmed up there, grew up on a small dairy farm, and started organic farming back in 1973. At one time farmed 500 acres in the 70s, working an outside job, and I got that down to actually farming about 25, and that was a goal. That wasn't that was a planned direction, and to raise some higher dollar crops. And we won't touch on that much today. So currently I have two areas we focus on, and one of that's local food systems, and the other is on water quality. So water quality issues do put me working with some of the larger growers and conventional growers that exist in this area.
Great. Thank you, Joe. And last but not least, Andre.
Yeah, I'm from Lenawee County, and I farmed for 35 years in Raisin Township with no-till. And when I retired, I became a director at the Conservation District in Lenawee County. Two other things that are, as I retired as an engineer. I believe in the proper use of land use underneath solar panels. And so as a small-time developer, I continue to stay close to that industry. And I lived in Germany for 3 years and watched their agricultural practices. And that was an insightful 3 years that I lived there.
Excellent. Thank you, Lee. And actually, Paul, you just joined us. Do you want to introduce yourself and where you're farming?
Yep. Paul and Valerie, we just bought 27 acres last year and are building a small, I'll say horse farm, but we're currently growing hay on the acreage in that. So we did that successfully last year, but we're totally new. I used to be in the steel industry, so I know zero.
All right. Got to start somewhere, right?
Right.
Okay. So we missed one of the first questions, but we got some good background from everyone, which is important. Why don't we just share sort of, if you have one practice that is your go-to conservation practice, what's something that you do consistently every year on your farm? And if you want to raise hands, that's great. I can start. So one thing that I do every year is I mulch my beds. Now I'm very small scale at this point, so I've always been small scale, but I keep my soil covered at all times, essentially. If it's not being used as a productive crop, it really retains moisture, it keeps weeds down, it builds soil over time. I mentioned to Joe before, it feeds worms too, which I'm really into. So Lee, you have your
Yeah, so I'd like to say the same thing about getting the soil covered. You know, I work when I was farming part-time, I worked full-time in the chemical industry. And the rate equation says if the temperature of an object goes up 10 degrees, the reaction rate doubles. And so anytime the sun shines on the ground, you can expect the humus to be mineralizing much faster. And a very big thing at the moment is how much carbon can we sequester in our soils, which basically is to reverse the last 150 years of mining that carbon. So my land's been no-tilled for a long time. However, I have a Napanine highly erodible on soils on one farm. I have Sphinx Hills on another farm, and I have Lenawee and Whiteville flat clay on a third farm. These are all small acreages, so I don't want to give the wrong impression. But when you think about cover crops and keeping the soils covered, I have like a collection of soils that exist in every single situation that coverage makes a difference.
we've got 30 seconds. Anyone else want to throw out? Go ahead, Mary.
I do composting. Everything I can get my hands on, I compost from my yard, from my kitchen. And I've done that since I was a very young child.
And I'll just note that we live on a creek, and we always make sure that all of our wetlands in our creek has a very strong buffer, and whether that's native or cool season grasses. There we go.
Wow.
Thank you, Lori.
It is great to see everyone.
That worked well.
Great. Okay. So we should all be back here. Did you all get kicked out of your rooms?
Mm-hmm.
Okay, great. So I'm going to ask, I'll start with Megan because I see her right here. We're going to have the moderator from each group just give like a 20-second high and low part about challenges and good success stories and challenges about conservation.
Well, we really just got to know each other a little bit. We've got quite a range of folks, of producers focusing on different crops, and folks who have some property, folks who are gardeners. So we just started sharing some conservation tips.
And that's great. And, you know, a big part of this is we just want people to get to know each other, know who does what around here.
Covering soil, compost, and buffer strips came out.
It's a great place to start a conversation. So what about you, Keely?
Pretty much the same. We only got to 2 of the questions, and the second one was really quick, but we talked a little bit about minimal till or tillage with a fork, in-bed composting, cover cropping, nuisance cover crops that turn into a nuisance and now they can't, so that was like a challenge for one of them. So it was just kind of getting to know each other as well. So
great. Hey, Doug, what about you?
Hey, yeah, thanks. So we had a good variety, a cattle farmer, hay and hops farmer, the Lenawee MEP technician, and a PhD at U of M in cover crops. So we did a little bit of introduction. We didn't get deep into the questions. So I think that if there's more opportunity to chat more, I think
Yeah,
our group
well,
is that. Yeah.
this is great. This is really just a way to get everyone to do another shop talk soon. So we got you. No, I'm just kidding. But anyway, I'm glad that everyone was able to just get to know each other. And we've got Nick also. Can't forget about Nick.
We had a couple producers on the opposite ends of the spectrum in terms of what they're doing. One really well established in terms of history, been farming for a few generations, and their family's been doing cover crops and conservation practices for decades now. And another farmer who's had leased their land previously and then was looking to operate it themselves and really are just starting to implement some conservation practices. So very two different starting points. So that was really interesting.
Yeah, it's interesting. It's, in this conservation world, we've been working in both the conservation district office and the NRCS office. We see people who are, the cool thing is there's like a level playing field. Everyone wants to be involved in conservation, and it just kind of brings together all these different pieces of agriculture. So that's the coolest part of being in this role. So we'll continue these conversations. I already am making a note that the next time we do a shop talk will allow more time for sure. So another part of today is we'll keep all that in mind. And we wanted to do this before our presentation because our presenter, Joe, may be able to kind of just talk a little bit about these things throughout his presentation. So, hey, Joe, are you here?
I'm here. Yep.
Great. So we've got Joe Scrimger. He's got a business called Bio-Systems. It's a little south of us in the Battle Creek Marshall area, but he's a wealth of knowledge in terms of organic farming systems on a larger scale and soil biology. So I'm going to let Joe have the floor for about 20 minutes. And remember, if you have any questions, just type them in on your chat and we will get to them afterwards. So Joe, you're going to share your screen. Let me stop sharing mine. Oh, look, I can see all these great people on here now.
What do I want to do here? Just leave it like that? We're going to wait for the slides. So I'll do it on that. Yeah. I think I'll just go ahead here, correct?
Well, you got to share your screen.
Yeah, but the slides come up. We don't want the slides yet. So which do I want to go to? Screen.
I can also.
I've got everything up but that. So.
All right. So what I'll do is I'll share my screen, and you can just tell me when to go to the next slide.
Well.
So let me hit.
No, I don't need the slides. We're going to do the narration first.
Okay.
So, and I've lost pretty much. Still there?
Yep. So you can go ahead with the narration, and I've got the slideshow up. So when you're done with that, you can.
Sort of lost the screen for a second here, but anyways, I'm going to go ahead, and then we'll get the slides after that. So sort of hang with me here because of time. I'm going to read the narration here to give you a little more on the history and the testing, and then we'll go through about a dozen slides, and I'll be able to talk a little bit more fluently with the slides. But this reading thing is it's just a matter to cover it in the time slot we have. So what we talked about with Ez early on was how we do a little different system of rating organic matter and water management and how we view tillage systems. So in this discussion this afternoon, I'd like to start with soil organic matter and rate a rating factor that was used in the 1980s called a humus chromatograph test that Woods End performed at that time, Woods End Lab. Having started running soil tests in the 1970s, I could see that some soils with similar organic matter levels performed differently. What this test showed was that whether the soil humus was degrading, building, or stable. So in simpler words, it meant that if a 3% organic matter soil was decreasing from, say, 4%, it would function differently than a 3% organic matter soil that was increasing from 2%. So in general, soil organic matter levels in Michigan have been decreasing over the last 100 years, which was mentioned in the breakout session, last 150 years, you could say. So roughly seeing half the organic matter lost in this time. In respect to soil nutrient release, if the soil is degrading, the nutrient releases decreases. Although in the short term, nitrogen can be released from the organic matter while the organic matter is degrading. So these soils will not hold or retain as much water as soils that are building or have adequate stable organic matter. So in the 80s, we found the Morgan test, which is a lighter acid test indicating nutrient solubility compared to a standard conventional, what I'd call a cation exchange capacity test, would also give a measure of how soil organic matter was functioning in relation to the amount of beneficial soil biology present. So after reading a book by Edwin Faulkner, The Plowman's Folly, in the late 1970s, I personally parked a two-way moldboard plow at our farm and purchased a chisel plow, used along with a disc, cover crops, green manures, to manage crop residues to promote adequate decay on heavier loam soils while improving water retention. The Plowman's Folly has been mentioned more recently in regenerative ag programs. I was also introduced to no-till farming in the 1970s while selling farm equipment from a company that had a heavy no-till planter. While doing some custom harvesting at the time, I was able to see the harvest results from moldboard plowing, conservation tillage, and no-till systems. But it wasn't really until 25 years later that I could say that conservation tillage with cover crops, green manures, longer rotations managed biologically, with respect to the beneficial soil microbes, really appeared to be the better system. In comparing longer term no-till corn, soy, wheat with some cover crop and tile to surface drainage, to surface tillage with a similar but longer rotation with hay and no tile, only surface ditches. In a 3-inch rain event, the conservation tillage system actually showed 2/3 less water runoff in heavier loam soils. So what I saw in properly managed organic farming systems also showed that they performed better in dry years at the 5-year point. In the Thumb Saginaw Valley region, even the bankers noticed that the organic farms functioned better in what was considered drought conditions compared to the conventional farming clients. Over a 25+ year period, the properly managed organic farms also show better in wet conditions, which was documented at Rodale Research Farm in Pennsylvania, their long-term comparisons of conventional and organic farming. What I could see and measure with soil food web test at the time was the increasing better measurements in beneficial soil biology on the well-managed organic farms. But farms do not have to be organic to improve the state and the amount of their soil organic matter. Excuse me for I should be watching you more than reading this, but we'll get it done here. While improving, so the amount of the organic matter while improving soil biology levels, which I believe regenerative ag farmers are recognizing in many ways. But the real successful regenerative farms are also marketing more of their process, similar to what organic farmers have done in the past, and not going to the commodity market, which improves their budgets for more conservation practices. So in bringing you up to more current dates, I would like to review 3 more recent farm models. The first being from Jackson County, raising corn, soy, and wheat on 5,000 acres, while more recently converting to 100% no-till on non-irrigated soil. What makes them unique is that they have utilized diverse cover crops on 100% of the land repeatedly, while also split applying 3 applications of nitrogen on their corn and wheat crops. This past year, they also custom grazed cattle on the cover crops at one farm. They do this while closely tracking the economics of the changes to maximize net positive changes to their operation. The second model is a corn, soy, and wheat cash crop operation on 1,500 acres in Lapeer County. They have done conservation tillage and no-till for many years, while working an outside job, and more recently are using vertical tillage on their ground while no-tilling the wheat and soybean crops. Vertical tillage on their corn ground while no-tilling the wheat and soybean crops. Some wheat cover crop is grown after harvest and burnt down in the fall. They have sold most of their other tillage equipment. They're on heavy clay soil, and they tile more ground and pay attention to harvest conditions. As they tile more ground and pay attention to harvest conditions, they say that no-till is working better. Because of time restraints, they don't split applied nitrogen or focus on other cover crops.
That phone call I had is.
You could, Connie, if you can, hey Connie, if you can mute your.
That's fine.
There we go. Okay.
The third model is also in Lapeer County and manages about 400 acres organically while working an outside job. They have had a more diverse rotation of corn, soy, sunflowers, oats, spelt, wheat, and clover without irrigation. They also had used surface tillage and started their operations in the mid-90s. What I want to focus on is an area that had been rented out in the past that had become a non-productive, depleted, very sandy, almost blow sand, roughly 5-acre field on a farm that was mostly heavier soil. Over a 6-7 year period, they built that field up with biological and organic soil building processes, including surface tillage, soil mineral amendments, compost, clover, and other cover crops, while going back to their whole farm rotation. The first year that it was corn was a drought year, and this field performed perfectly. No yellow leaves, very green and productive corn, while conventional corn in the area, even on very productive black sandy loam soils with beef manure, were burning up in the drought. So the point I want to make with this field is that I keep hearing in regenerative ag and soil conservation programs that tillage is the problem. So, and that contradicts what we did with the sand. But what I have seen over the last 50 years is, yes, too much tillage can be a problem, especially with deep moldboard plowing. But the larger problem, from my viewpoint, tends to be that too much elemental nitrogen burning up soil organic matter and decreasing the state of the organic matter to the point that it becomes more mineral ash than humus and does not have the ability to hold itself against wind and water erosion. In general, I and other researchers have concluded that the soil can only hold about 70 pounds of added soluble nitrogen at any one time. Sandy soil is a little less, clay soil is a little more. And I understand that corn may need more than that. So if it is split applied, the soil can still function and release nitrogen on its own. Feed the soil too much nitrogen and it quits producing it because it senses there is too much, which is harmful to the soil and the plant's health that is growing on that soil. So farm model number 1 is splitting their nitrogen application, intensively cover cropping while starting to reintroduce animals, while monitoring the economics of these changes. Farm model number 2, I think, could learn from farm model number 1. If split nitrogen and cover crops pay, then he could afford to have the applications custom done, especially if his soil improves more in the long term. I also see both of the first 2 models currently starting to apply more fungicide, while farm number 3 does not have fungal disease issues. Farm number 3 also makes and utilizes compost as a good part of its soil building program. Possibly the first 2 more conventionally marketed farms could learn some from farm number 3, even though it is an organic farm. Understanding that a good working 3% organic matter soil has the potential estimated nitrogen release, what is called ENR, of around 100 pounds. And that's according to A&L Labs. Before a legume, animal, or green manure, nitrogen credit in the rotation is taken. So if soil organic matter is in a good humus state, cover cropping is done, along with manure, compost, or green, it is possible to have 250 pounds of nitrogen available in slow release form before you buy the little that may be needed to get the crop started in wet, cold conditions. So with the soil food web test, we can identify and balance the soil bacteria and fungal organisms while monitoring and building beneficial nematodes that will suppress plant feeders while improving nutrient utilization. And if we build beneficial soil fungal organisms, we not only get better phosphate utilization for early growth, late season maturity, and quality, while getting drought and disease resistance. Now that's the dynamic soil process working for the farm. The key is in managing the beneficial organisms, not in waging war on pests. While using today's biological science and technology, not just chemical technology based on outdated science, to truly become a high-tech farmer. While having faith in investing in and protecting your soil, organic matter, and biology, it truly pays. So with that, I'll bring up some slides and try to get back to a little more relaxed presentation here.
Hey, Joe, so what I'll do is I'll just let me know when you're ready for the next slide, because I've got it pulled up already.
Wait a minute, I've got it. Let me do it so I can do the,
okay.
Can
so
I
go ahead
do
and
it?
share your slide?
I've got it here.
Okay.
So let me see here. So has everybody got it?
So you got to hit share on the bottom.
Okay. Now I can't get there. So
all right. So what I'm going to do is
just a second. I don't have that up. So you got to do it. Yep.
Okay. So
I've lost the whole thing, so I don't even have your screen.
Okay. So
technical. Just a second. So I'm back here to Zoom. So what did I lose?
So we're at the rating or the first slide of your presentation.
Yeah, and I don't. Okay, I have your chat room. And so how am I going to get back to the regular room?
So all you need to do is pull up your presentation and tell me that we'll be on the first slide.
So I'm going to pull it up and go, but I won't be.
That's fine.
Deciding. Okay. Okay. So first slide. As we progress there, again, we're rating organic matter, water management, and tillage systems. And when we get done, we might want to come back to this farm that's showed in the background. That's organic spelt in the foreground, 100 cow dairy. It's doing composting, a lot of cover crops on super heavy ground in the center of the state. So we might want to come back to that at the end. So next slide, please.
Yep.
So biotic soil components are very important. And this is from field crop ecology. There was a doctor by the name of Richard Harwood that worked at MSU quite a few years ago. He recently passed away. Very nice man. We worked with him on this book, a group of us. And this I felt is one of the more important pages of the book. So at the top we have plant roots, which you all know about and we're all learning more about. And learning it's not only what's above the ground, it's what's below the ground that's important. And on the bottom you have earthworms, and which are very important, and we're getting a better respect for them. But in between we have this bacteria, which we're learning more about, and now we've got these microscopes that we can pick them up with too, and you know, for $250 you can monitor those, you know, at the farm. We got fungi. We got Oceaniomyces that give that soil that smell that you always have when you go to the woods and take the smell, but you don't always have in agriculture of today. You've got nematodes, which most farmers think about harmful plant feeding nematodes. And that's the problem of focusing on the bad guys versus managing the good guys. If you're managing the soil properly, you'll have more beneficial nematodes than you'll have plant feeders. But whenever there's a rule here that I want you to remember, whenever you decrease the overall numbers, the unbeneficial always go up. Whenever you increase the overall numbers, the unbeneficial always goes down. And that's relatively across Mother Nature. It's not just about nematodes, but nematodes are the thing that we've really documented in this state, and we can do that. So nematodes are a measure of soil quality with me. So we got protozoa, again, takes the microscope And then we got those arthropods that are much bigger that you can see that are working for you in the soil. So next bullet there.
Yep.
So the next bullet, the bacteria more deals with the nitrogen. Next is the fungal, yields the phosphate, and the mycorrhiza yields moisture and phosphate efficiency. And there's really a lot being said about them now. We've been talking about them for 25 years. And these nematodes is being soil quality. So next slide. Next slide, the soil food web analysis is one of the things we've worked with for the last 25 years. And we're really fascinated with Lane Ingram. And she was from the University of Oregon. And I met her up at Sutton's Bay, working with the fruit project up there. But so in this slide on the top green area, we've got active bacteria and total bacteria. So they're measuring the dead biomass of bacteria and the live ones too, along with the fungal, dead and alive, protozoa, the flagellates and amoeba are more the beneficials. As you have more silates, we can tell that soil's gone more anaerobic. The mycorrhiza colonization, endo and ecto, sort of close to the root or in the root and outside the root. And typically that's between the roots and the soil. That's working for the plant extended root system. It's making that plant's root system much more dynamic, especially when you get the numbers up. And then this balance of bacterial and fungi. Most farming soils are bacterial dominated, and they need to be a balance of bacterial fungal. A lot of times we need to build the bacteria, but we need to respect the fungal, and we need to cap the bacteria, and most farms aren't doing that because they're too focused on nitrogen because of the poor quality organic matter. Then on the right, we've got nematodes, and we really need to look more than 3 or 4 varieties. We need to have the whole spectrum. Next slide, please. So just a little history on the testing, and you can see this test is from up at the top back in 2007. And so, and the test on the left is labeled A&L lab in the vertical column there with the 1% highlighted. And the test on the right is called a biotest, but that would be an adapted Morgan test that we used. Currently, I use a company called International Ag Labs that run the Morgan test. And we still use the CEC test, whether it's International Ag Labs or I sort of prefer A&L because they're the ones I've worked with for the last 40 some years. But if you can see the situation we had here is a problem. If you'd have looked at the A&L labs, you couldn't really pick up there was a problem in that soil. You got a good pH, you've got high phosphorus, P1 high, but not as high as some, but low organic matter. Everybody would pick up on that. But like in this Battle Creek Marshall area, they'd consider that average organic matter. It's like back in the thumb, we'd work around 2.5, 3% organic matter, and see some areas in the Saginaw Valley up to 4 or 5, but we see some areas in the Saginaw Valley that were down below 2% too. But the bigger thing here, when you look at the soluble phosphorus in the middle on that biotest, or what I'm going to call a Morgan test now, we had 35 pounds releasing out of 377. What that represents is there's basically no fungal organisms beneficial at all. That situation is set up for fungal disease, meaning whenever the beneficials go down, the non-beneficials always go up. And you got that situation, and you can tell it because of how the minerals are releasing. So we use that mineral test as an indication of biology because it was a $25 test versus a $250 biology test. We didn't run those soil food web tests very often. But more importantly, now most all soils we picked up early on this, they don't have enough bacteria working either. They won't get their calcium soluble. So they have a 6.2 pH on the left, 700 pounds of calcium. But on the right column, they had a 0 to 150 working. And the soil in a fresh, moist state, which we always run those samples on a as-is soil state. We never dry them. It actually showed up a lower pH, meaning that soil was acting acid. And that's the end result of some of agriculture today. The soil acts acid, and it doesn't release the nutrients the way it should. So next slide, please. Or next bullet, I guess, for me. Phosphate, we got to get it soluble. We got to get the calcium working. But if in this soil we were decreasing nutrient uptake, we had a potential for weed pressure here, and that would be quackgrass right off the bat. And quackgrass tries to correct that situation. Once you get to view the weeds, is what they're telling you about the soil, it's a whole different situation than which herbicide to apply. You can actually deal with the cover crops to actually control the weeds. We'll talk about that a little more then. So hope you got the cover crop slide up. I probably told you next slide.
Yep.
The green cover, is that one up as?
Yep.
Okay, so, and as we hit the bullets there, we get this allelopathic effect from the cover crop. But as a farmer, no matter how big you are, when you harvest a crop, and whether you're tilling or not, you're obligated to turn that soil green again. And if you keep it green, Mother Nature won't have to keep it green. And that's about 70–80% of the weed problem, is we haven't kept it green, so Mother Nature brings these green things in. And then sometime you have the thing about, I call the dog bane effect, which is, you know, you use a herbicide to control milkweed, and it's very easy to kill. Then you get dog bane, and dog bane is very hard to kill. And I'd rather work with milkweed. But if you can correct the compaction layer in the soil down where that root is going vertical, and milkweed always has a vertical root, and so does dog bane. But when you correct that compacted layer, and you have to do it biologically, not just mechanically, then you don't get the milkweed anymore. So, so your responsibility to keep it green and broadcast cover crop either into a growing crop for the no-till guys, or lightly disc for some of the organic people or conservation tillage people. So next slide is green manure and cover crops. And as each section comes up there, all farms we like to see, especially when they start this biological agriculture. And actually I did organic farming, but I work with farmers on a three-way basis. I work with organic farmers, I work with biological farmers. Some of them are transitioning to ag, and we think it's a transition. You don't just go cold turkey. You systematically go through this transition so you don't lose yield. And so so we got organic farmers, we got biological farmers, we got conventional growers. Oats is a cover crop. We use it to help metabolize phosphorus. It's really unique. Oats grow in cold weather. Corn and wheat doesn't. And that's because of how it can metabolize phosphorus and put it available back into the system. Buckwheat, that's my real go-to summer hot cover crop. When nothing else will grow, it only takes a little bit of rain. 3 weeks, you got buckwheat up and blossoming. But buckwheat brings the trace minerals back into the system, really a unique crop. Then you got all that crop residue that you want to manage it back in, because in those root systems, as you manage that residue back into the system and get it to decay properly, you recharge your potassium. You just don't have to buy as much. So next slide. Organic matter in the state. So we talked to you about that, that humus chromograph test. But basically what that humus chromograph test is telling you, whether you've got anaerobic soil. Number one problem in North American agriculture is soil compaction. That's some degree of anaerobic soil. And when you got anaerobic soil, that digestion that goes on converts the organic matter to ash, but ash will still test as organic matter. So you can't tell that it's digested the wrong way, but the soil gets harder. If you deal with enough Amish farmers who plow with horses, they understand this. So because their horsepower doesn't cover up the fact that the soil gets harder. Even when you run manure systems, they understand the difference in manure systems. You work with anaerobic liquid manure, they worked with it for 20 years and they quit working with it because the soil gets harder. You got to have an aerobic digestive system in your manure, even liquid, and that can be done. So aerobic digestion, but it wants to be controlled aerobic digestion. Not controlled is burning up organic matter, or not controlled and unlimited is fire. We don't want that much. We just want controlled aerobic digestion, and it'll yield humus, whether it's in the soil or whether it's in the compost pile. Build organic matter, building beneficial organisms. Most soil beneficial organisms are aerobic. Anaerobic are more disease causing. They really need to understand that, especially in the times that we're in. So this nitrogen thing in relation to this nitrogen release in organic matter. So this is just a chart that actually used to be published on the back of A&L soil test back in the 80s, and they don't do it anymore. So they used to put E&R on the front side and then this chart on the back side. But so a 3% organic matter soil has the average in clay or sandy soil to do you about 100 pounds of nitrogen. The thing is, they quit publishing that because most soils aren't yielding 100 pounds of nitrogen anymore. And they didn't want farmers to think that they were going to. The key is if the soil is functioning properly, it will release that. And I did nitrate tests for 40 years to try to establish that consistently. But you can't tell much from one nitrate test. You have to do multiple tests to get the soil figured out. But by the time you add your legumes and animal manure, your compost, your green manure, and cover crops, you can come up with that 250 pounds of nitrogen before. So if you are conventional and you're doing a real long day corn, yeah, you may still need to apply a little bit, but not very much. Next slide, please. So this is that model number 3 farm we talked about. And as they put the bullets in there, it was conventionally farmed out before. It was blow sand. It was set aside. And this is after he put it through the building process. And I know you've all seen corn. The thing is, this is in the middle of a drought. And that corn showed better than the corn on the heavy ground. But it also showed better than the corn on the black sand that was getting beef manure. And no irrigation. And it typically yielded good after we got it back on track. It took 5 years of building to get that back on track. So it was put in, but it started out as a biological approach, and we're going to talk to you about what that meant. So the procedure over the next slide, procedures, you got that one?
Yep.
Okay, so the length of time, first we started out doing tests. We did these bio tests, which is an adapted Morgan test. We did observation. We smelled the soil and see where we're at. And we didn't have much smell on the start. Did some other tests, but we're not going to talk about that. Did the rotation and did crops and green manures, but we did a buckwheat crop and a rye crop, a clover seed crop. We did surface tillage. While we were doing these amendments, in the first year, because the phosphate was there and not working, we used some MAP. No, don't overuse that. That'd be 1152O. It's fairly hot phosphate. It does raise the energy, and if you use it right, it's not hard on the soil. It's hard on the soil if you use too much. We used ammonium sulfate, but after we limed to help release the lime and keep the energy up in the soil, we used compost, which poultry compost, medium carbon compost, and high carbon compost. And you really need to understand the 3 types of, there's multiple types, but we break them down into 3 types to know what we're doing. And in the long term, actually, this high carbon, low testing compost is really one of the more valuable things to your soil. We used high calcium lime, dolomite lime, not excessive amounts, light rates. We used some gypsum. We used Calphos, which is a slow release phosphate. We used some sulfamag because it's lighter, sandy soil. Use a little bone meal. You don't use bone meal unless you can buy it in full semi-loads to get a right price. And so we didn't put semi-loads on here. They were distributed to other farms that needed it. Bone meal is still one of the best things for raising roses if you want to get the color in them. But that color that vibrancy you want in all crops, not just roses. And we applied some boron here too. So next slide, please. And this will be the last slide. And we want to show you what happened over time. If you can see, we got a 1996 test. Now we got 2 types of tests integrated in through here. We don't have time really to explain it all. So I'll just explain what I can, and Ezra will stop me when I can't. So you can see in 96, we were at 1.4% organic matter. And that was after we already worked some with it, started at 1%. And then it went up to 2.3%, which it should have went, you could have went up more if you more intensively did it. But we were building, and as you go down at the second yellow line, we started out with that 35 pounds of soluble phosphorus. We ended up with 175, and that's soluble on the Morgan test. The reserve was at 189. That's your typical phosphorus test, and that's in pounds per acre of phosphate, not phosphorus. So understand, it's already been converted from parts per million times 4.6 to get there. It's relatively stayed the same, but our solubility totally changed. Our reserve changed partially because of the compost and the Calphos application. So we did have a higher reserve on the P2 phosphate. Potash stayed the same. The big thing on the calcium, starting out with 0 soluble calcium, we actually went up to 5,600, but that test is hard to quantify because there was only 1,900 pounds totally there. All 5,600 pounds says is that it's very highly soluble, that you're not limited by calcium as long as you don't overfeed other things like nitrogen or potassium, because either one of those will interfere with calcium uptake. And that's why a lot of farmers end up applying more calcium, because they have too much nitrogen or potassium in the system and it interferes with other mineral uptake. So I think that's basically, we changed the potassium balance tremendously, and it still needs to be changed more here for high quality production. We don't want to go with excess potassium. Potassium is the easiest nutrient in the soil to release, and you want soil working so it releases. And then you want to keep it keenly balanced. You don't want to go try to do too low. You can't get a good yield at too low potassium, and you can't get good structure in the crop. But also understand, potassium does the structure, that's that structure in the stalks. But also things like silica also do that structure too. And don't forget about the silica. So last slide ends it. And then what I want you to do is just go back to the first slide.
Sure.
So if you can,
We'll have about
show
1 minute left here.
Excuse me, 1 minute left.
Yep.
For questions.
We are back on the first
What
slide.
your questions and go back to the first slide while they're getting their questions to me.
Okay. And what we'll do is, if you do have questions, you can also email them to me because we're a little short on time.
Yep.
And we'll make sure that we get that to all of you. So that was quite a bit of information, but it was also very, very informative. So we're very appreciative of that. And it's good because we have a lot of different types of farmers here. I think a lot of that certainly speaks to us in terms of the tillage and keeping, you know, if you have a, like you said, if you turn the soil, you have a responsibility to kind of bring that back, make that green again. So I really appreciate it. What we're going to do now is just, if you do have questions, just let me know. You've got my email, and I'll put that back up here in a second. So let's see, still got my screen shared, but feel free to send me an email. I will put that in the chat. My question for everyone is, what is a Shoptalk event without door prizes, right? So we got door prizes. So Megan will get a hold of you if you want a door prize. There's gift certificates to the Dexter Mill and to Ann Arbor Seed Company. We got some other stuff, and also Megan just sent you a survey to everybody, so make sure you do the survey. That'll be helpful feedback for us. And I know that it was a lot of information, but it was also very informative, and we try to do best we can with 1 hour. I think we did the best job anyone could do with an hour. So we got to know everyone. We got to meet and greet. We got to learn about organic matter, tillage, and water systems. So that is about it. Let's see what we got in my chat here. So we will also post this recording on our YouTube page. So if you want to revisit the slides, you can. And I think I'll be able to send you the slides as well. So make sure you do that. And I thank everyone for tuning in. And I thank Joe for the great wealth of knowledge that he shared with us today. So
can I just say a couple things here?
Absolutely.
I appreciate it. I should have really caught it when we were in the breakout sessions. That time was going to go that fast. I knew it was.
Hey, that's all right. We still got a minute.
So the question I'm going to ask you, and I want you all to think about, is what is the state soil? State bird's a robin. So get back with me on when you find out about the state soil and what happened to it. So
I think it's on that note. I think it's pretty crazy that we all know that the state bird is a robin, but who knows what the state soil is. So I guess I won't ruin the game. But it's not common knowledge like the robin. So, you know, you learn in school that the robin is a state bird. You should learn the state soil. So, yeah, thanks again. And we thank everyone for coming, and we'll be in touch with all of you soon. So have a great day.
Great.
Look forward to seeing you.
Very much.
Thank you.
Thank you. It was great. Appreciate it.